Hey everyone, today we're going to talk about Exploration 3 from Lesson 1. So on your books, I'm on page 131 right now, and this is a little bit of a review, but it talks about mutation. So this was talked about in the hands-on lab. We didn't do that hands-on lab in class, but we're going to talk a little bit about the mutation part of it. So sometimes DNA can change, and that's called a mutation. Okay, so mutations can happen in a lot of different ways. Sometimes they're random errors. Sometimes they can be caused by environmental factors. Radiation is a big thing that causes mutation in DNA. So mutations can be beneficial, they can be neutral, or they can be harmful. So they fall into one of those three categories. So a mutation may have a benefit. It may be beneficial if it promotes resistance to a disease or a toxin. So in that case, that would help that organism survive, and that would be a benefit or beneficial mutation. They can also have harmful outcomes, such as causing genetic diseases. And so in those cases, those would be negative mutations and those would be harmful mutations. And then a lot of mutations are neutral, meaning they don't really have an effect on the organism, on its ability to survive and reproduce. So some of these things can be like in eye color or hair color. They don't really affect the health of the individual. They don't affect whether that organism is going to be able to reproduce in the future or not. And so those would be kind of these neutral mutations. They don't really matter too much. So a lot of mutations can be neutral. A lot of them, if they're a neutral mutation, we may not even realize that there was a mutation at all. Okay, so suppose that a gene mutation does not affect the protein that the gene codes for. Now, what effect would the mutation have on the individual? Well, in this case, if the mutation does not change the protein, then there should not be an effect on the individual. This might be a neutral mutation in these cases. That might be a neutral mutation. Okay, so as we look at Exploration 3, if we were looking at plants here, say we're looking at plants, there's many things that we can study on those plants. Okay, so in all living organisms, we can look at traits, and traits are passed on from their parents to their offspring. Some traits can be learned. Okay, some traits can be learned. Walking or riding a bike is a learned trait. We're not born with the ability just to ride a bike. That's a learned trait. But some traits uh, are you're born with, or they are inherited. Okay, so let's say if we were to look at these plants right here, and we had a horticulturist that was constrained by time. They wanted to know what traits these plants had, so they could just breed these plants over and over and over until they got just the one trait that they wanted. That's what Gregor Mendel did. But in this case, if they're constrained by time, they need to know it a little bit faster. And so what the horticulturist could do was send the seed plants or specimens in to a lab for genotype analysis. And so it would actually tell what the genotypes of those plants are. Okay, it usually takes a couple weeks. Humans can actually do the same thing. That's kind of what happens if you ever send your DNA sample into like Ancestry, DNA, or that 23andMe, those companies that actually provide that service to humans. Okay, so genes are passed from parents to offspring. So genes come from your parents. So half of your genes came from your mother and half of them came from your father. And your mother and father have the same number of chromosomes, and on the chromosome you have the same genes and the same locations. So these two pictures over here are two different chromosomes. Like one of them came from the mother and one of them came from the father. And they have the same genes that are in the same locations on that chromosome. So this is a whole bunch of DNA on a chromosome. And then one section of that DNA might be one gene. Okay, so in this case, the mother and the father, we have the mother that has a dominant gene for whatever this A is coding for, and then the father has the recessive, the lowercase. But then the B, they both have the same. And then it's the opposite, the recessive for C for the mother and the dominant C for the father. And so they have the same genes in the same locations on chromosomes. That's important to remember. Same genes and same locations. Okay, since half the DNA comes from the mother, half comes from the father, this is how your genotype is made. It matches up the gene from the mother and the gene from the father, and that becomes the new organism's genotype. So Mendel did that with pea plants. So Mendel crossed pea plants and created different hybrids of pea plants. And so he looked at different traits, but one of the traits he looked at was the color of the peas. Okay, so peas can either have yellow, they can have green, well, and that's really it, but they can also have other traits such as round or wrinkled, and the flower colors can be different, and the heights of pea plants can be different. But what he looked at, one of the two, was yellow pea plant, the yellow peas and green peas. And what he found was that the genotype for peas, if it's yellow, is dominant to green. And so if you have two dominant alleles there, you're going to have a yellow pea. If you have two recessives, you're going to have a green pea. And if you're one of each, you would have a yellow pea. Okay, so the offspring inherits one allele from each parent. Okay, and how we test this is through a Punnett square. How we test this is through a Punnett square. So if we look at this Punnett square, this is pretty simple. We kind of did this with that Penny Genetics lab, but we take a genotype that's at the top. So what we're crossing here in this particular Punnett square, we are crossing one organism that has a genotype of capital F is being crossed with another that has lowercase f. So we take these two capital F's, we bring them over to one side, we split them apart. Two lowercase, we bring them to the other side, split them apart, and then you just match them up together. So we take this capital F, we bring it down, capital F, bring it down, lowercase f across, lowercase f across. Same thing over here. And what we see here is we use the laws of inheritance and the predictions in the Punnett square to explain how the genetic factors can influence the growth of organisms. So the inherited alleles determine an organism's traits. These traits, which are also called genes, can affect the growth and development of an organism. So your traits affect the growth and development of you. So many traits, such as fur color, are determined by more than one gene. So how might the influence of multiple genes affect the number of possible phenotypes for fur color? And so there might be a greater number of phenotypes for this. So if we have lots of genes that determine a trait like fur color or hair color, there might be many different types that we could have, many different variations. So the more genes that affect a trait, the more different variations that we can have. The same is true for humans, like in height. So there's many genes that determine a human's height. And so that's why we can have humans that are many different heights. Okay, there can also be environmental factors that play a role in that. So if a human has, as they're growing, has maybe some poor nutrition, they may not be as tall as what they could have been if they would have had better nutrition. So if we look here at this Punnett square, if we complete this Punnett square, again, we are crossing two heterozygous. So capital R, lowercase r is being crossed with a capital R and another lowercase r. So if we put these on the side again, so this one goes over here to this side, this one comes here, and we just match them up again. So you bring it down, bring that capital R down, capital R across, lowercase r across, and we can get these different genotypes. So if we look at our chart here, the capital R genotype, the probability was 1 out of 4. So 1 divided by 4, that'd be a 25% chance that we are going to have that. The capital R, lowercase r genotype, well, we have 2 out of the possible 4, and so we'd have a 50% chance of that one. Lowercase r genotype, we have 1 out of the 4, so I have a 25%. Red feathers, so the capital R is dominant, so anything with a capital R is going to have red feathers, so we have 3 of those boxes have capital R, so 3 would have red feathers, that's 75%. So, and then finally, brown feathers is our recessive trait. So the only one that would be brown is the one that has 2 recessive alleles right here. So that would be 25%. Okay, so we're going to do a lot of Punnett square practice here today, guys. So as we work on that Punnett square practice, you can refer back to this, and this will help you maybe get started on your Punnett square worksheets that will be assigned this week.